Hi everyone. In this video, we will be talking more in regards to cell structure and function. However, in this video, we'll be particularly focused on the plasma membrane. So, what is so important about the plasma membranes that they are requiring an entire video of its own? It's basically the membrane or the outer layer that separates life from non-life. And several functions of the cell membrane itself include separating the cell's interior from the cell's external environment, keeping damaging materials out of the cell and allow entry of needed materials that are needed in the cell and facilitate the chemical reactions that are critical for life itself. And then it also receives external signals and initiate any cellular responses and basically adhere to any neighboring cells or the extracellular matrix, or what is called the ECM. So when remember when we were talking about phospholipids, where when they go into like water, there's like a hydrophilic area and a hydrophobic area for phospholipids. When they're placed into water, what happens is that they form little membranes where it's like either this shape if it's one singular layer, and it's like a one single layer where it's like this. That's called a micelle. It's a single layer phospholipid membrane. And then we have this type of format where it's like two layers. So it's like bilayers. And this is what we would commonly see with cell membranes. So the region between the plasma membrane, the outer membrane, and the nuclear membrane is called the cytoplasm. And this basically consists of organelles that are suspended in the gel-like cytosol. So the liquid itself is called cytosol. And then it also contains the cytoskeleton itself. 70 to 80% of the plasm itself is water, but it has semi-solid consistency depending on the proteins that exist within it. So the eukaryotic plasma membrane itself is a phospholipid bilayer with many different either associated proteins or embedded proteins that you see here. It can be also cholesterols, different lipids with carbohydrate attached, like glycolipids or glycoproteins, so on. So now, what uh, uh, so one thing about the membrane itself, it has to be stable. If they are too fluid like this, where there's too much space and like things going on, it would basically melt. If we have something that's too solid like this, where it's viscous, it will end up being too rigid, end up breaking. Now, there are many factors that affect membrane stability, and this includes the saturation level of the fatty acids, where if it's more saturated like this, it ends up viscous, and if it's less saturated, it ends up fluid. The presence of cholesterol can also affect the stability itself. If there's more cholesterol, it's more stable. If there's less, it's less stable. And then the temperature itself can play a role in membrane stability, where if it gets colder, it's more viscous. If it's um, warmer, it's a lot more fluid. And what this kind of led to, all the stability ideas was giving towards the idea of how the membrane itself is set up. And this aspect is called the fluid mosaic model. It basically just states that the membrane itself is contained of a mosaic of components where it includes like phospholipids, cholesterol, proteins, and carbohydrates that move past each other and give the membrane a more fluid character, where it's not too fluid that it melts, or too rigid that it ends up breaking with one movement. So now with cells, they have to connect with each other. So there are several ways in the way that they connect with each other. And some of these includes intercellular junctions, where they provide direct channels of communication between cells. The cytoplasm, the ions, and the signal molecules can move from cell to cell. And depending on the type of cell, whether it's plants or animals, they do this completely differently. Where plant cells have like plasmodesmatas, like this, or gap junctions within like animal cells. So there are a couple of intercellular junctions that allow all of this interaction to occur. And the first one we'll talk about is a tight junction. A tight junction is basically a watertight seal between animal cells that prevent any leaking between cells. These are typically found between the epithelial cells that you see here, like these, and uh, basically the ones that line your internal organs and your cavities. The next one is desmosomes, where they are short proteins in the plasma membrane, like adherins that act as spot welds, so like these here. And these end up joining adjacent cells in tissues that stretch, such as like hearts, lungs, and muscles, because the heart is beating, the lungs are beating, the muscles can stretch or contract depending on what is happening. And these are only present within animal cells. So now one thing about the membrane is that it has selective permeability, which means that it has a tendency of allowing certain substances to pass through. And with phospholipid bilayers, they have a selective permeability, in that the hydrophobic ones, or the nonpolar ones, move across quickly. Small uncharged ones can also cross, but it's a bit slower. But then uncharged ones, or large polar ones, or ions, they uh, cross very, very slow, if they even get through that membrane itself. So what actually affects membrane permeability? It's more affected by it's increased when it's like more unsaturated fatty acids. So if the lipid bilayer itself has more of the unsaturated fatty acids, what it happens is that they can just like go through. If there's any like short hydrocarbon tails, any less uh, uh, cholesterol, and warmer temperatures, that it's like letting loose a little bit. It decreases permeability when there's more saturated fatty acids, long hydrocarbon tails, more cholesterol, making it a bit more rigid, that it's not allowing anything to go through, and colder temperatures as well, which means it has less fluidity compared to the other sets. So now for membrane transport. So now that we know how the membrane itself is permeable and how it is having many different factors, we still need the molecules that are like a large polar ones that are critical for like a different biological processes. So the way they do that is through different forms of membrane transport. And three of them include diffusion, facilitated diffusion, and active transport. And what happens here is that with this, the first two are passive movements, in that it does not require energy for the molecule to go through. In active transport, it actually requires energy like ATP or like an electrochemical gradient that allows it to go through. So what actually drives diffusion? So our molecules, any molecules, any ions, they have what is called thermal kinetic energy, which are basically in constant random motion and just like hyper bouncing off the walls kind of scenario. But when there is like a concentration gradient, what happens is that if there's any high level concentrations in one area and a low in the other, what they would try to do is try to get it to an equilibrium where they are in equal um, uh, concentrations. So that's what basically drives diffusion itself. It basically, so simple diffusion itself occurs when it goes from an area of high concentration to low concentration. So in like membranes, this occurs through the lipid bilayer. If there's like more here, none here, it just starts going through, and what happens is that it reaches that equilibrium where it's balancing out the sets of concentration of this molecule or substance. Usually this is for non-polar small molecules like O2, CO2, any lipid hormones, so on. Now, we have a specific case of diffusion that is specifically for water. That is called osmosis. And what happens is that the solutes themselves cannot really cross over, but water itself can keep moving through the membrane itself. So if there's less water here, but more solute, what happens is the water redistributes itself to bring it back to equilibrium. So it would go from uh, uh, the water would go from an area with uh, less solute to more solute. Now, there is also a terminology referred to as osmolarity. It basically describes the total concentration of a solute in a solution. So this would have, in this case, when solutions with this different osmolarities are separated by like a membrane here, what happens is that it ends up with the water with the lower concentration moving into where it's higher concentration of the solute. So it reaches out that balance with the amount of solute in the amount of water that it is. So it goes from lower osmolarity, so more water, across the membrane to the area with higher osmolarity with less water. So this here would be considered something that has less less osmolarity because it has more water, and then this one would have a higher osmolarity because it has less water. Now, when we think about all of this, it also kind of brings in the idea of tonicity. Tonicity is basically describing how the extracellular solution can change the volume of a cell by affecting the osmosis through the selectively permeable membrane. Basically, how if it would basically be based on 3 different conditions called isotonic, hypotonic, and hypertonic, which means that if you put like a this where the where the solution outside has the same amount of solute inside the cell and outside the area, it will remain comparable. But then when we have a hypotonic situation where the solution around the cell has less solute compared to the inside and more water, like this case where it's like pure water, what happens is that all of the water goes into our cell and ends up with a cell that swells to a point where it can burst. And then when it's hypertonic, where the solution outside the cell itself is highly concentrated with solutes, it ends up with the water wanting to come out of the cell, that it ends up shriveling up to a point where it's like this. And most cases, animal cells end up functioning best when the extracellular fluids are isotonic, and we like to keep it that way. So these are some a little bit of a review, kind of just explaining what I've just explained, where hypertonic solutions, the water moves out of the cell. Hypotonic solutions, water moves into the cell and then basically becomes turgid or burst, depending on the type of cell it is, if it's plant cell or an animal cell. But then if it's like isotonic, it just stays okay. It's a happy cell at that point. Now, what we need to kind of discuss is what is called osmoregulation. So in most cases with plants, fungi, and bacteria, what happens is that bacteria and like certain protists, they prefer a hypotonic extracellular solution, like pure water, basically. They like to be all happy with all the water in them. But as the water enters the cell, it expands to a point where the membrane is pressing against the cell wall itself. So basically turgor pressure to a point where it becomes turgid like this. It's a happy plant cell. Now, when it comes to turgor pressure, it's very critical to actually keep the organism's growth and function being proper. And if it goes into a hypertonic stage, what happens is it causes an irreversible process called plasmolysis. And this is not good, considering it looks like this, where it's like all shriveled up inside, where it's getting a bit shrunk down. Now, when it comes to active and passive mechanisms of membrane transport, some of these can occur during using membrane proteins. So we have regular diffusion, which doesn't require any um, uh, membrane proteins. However, with facilitated diffusion and active transport, what happens is that you need a membrane protein to assist in going through the membrane. So with facilitated diffusion, there is no energy required, but then with active transport, you do need energy. In some places, they ATP, which is like the energy source, and it binds to it to kind of make it more flexible for whatever component needs to come in. So like in this case, it would be like sodium ions or potassium ions to kind of go in and out easily. So now some of these membrane proteins, they are basically some proteins that are embedded or attached peripherally to the cell membrane. And what is seen is that we have like two different types, where one are integral or transmembrane membrane proteins, or peripheral membrane proteins. So these transmembrane proteins are amphipathic, so they have like they basically go through the exterior and interior surfaces of the cell. And then peripheral membrane proteins are only on one side of the membrane, so they don't span all the way through like integral ones or transmembrane ones. And it can be in different shapes, sizes, so on. So just keep an eye out for like these examples. And what happens is with transport proteins that are transmembrane proteins, they end up transporting the molecules through the cell membrane. And three broad classes of these are channels, like these, or carrier proteins, like these, that are like allowing them to come in. And then some of these like that are pumps. And depending on the type of transport protein that exists, it has an effect in membrane permeability. So next, what happens is we have certain ion channels and an electrochemical gradient. This is basically allowing the flow of ions and small molecules through the membranes. And this is very controlled. That way it doesn't send it into a reflux of ions that may be toxic at that point if there's an increase in any of the ions. So ion channels specifically allow the diffusion of ions down the gradient. Now, electrochemical gradients, they occur when ions build up on one side, establishing a gradient by concentration and electrical, so charge. So if there's a high charge outside, it wants to go inside. So it will go down the gradient itself, so like this here. So if there's more sodium ions here, it would have a high concentration and a net positive charge, where it wants to go high to low in terms of the electrochemical gradient. Now, these channel proteins are strictly regulated through certain things called channel proteins. They basically open and close depending on the response from any signals that are outside. For example, if there's like a binding of a particular molecule like this, like these signals here, it'd be like, okay, we need to get things going, or it closes again once the condition is reversed. So once the signal molecule or the ions or any kind of pressure comes in, it opens it out and then lets the ions through. And then once everything is at equilibrium, what happens, it let goes and closes it again. Now, these channel proteins can be very selective, despite it sounded like. So one of the things that they do is that with these channel proteins, what happens is that the structure itself permits only a particular type of ion or small molecule to pass through it. And certain particles can only pass if they're like the correct size to them. So like for these, it's like aquaporin. It's like certain thing, it's like a pore for like water molecules to go in and out. So it's one of my favorite channel proteins. They let water in and out without having any issues, but it has to be the water molecule that goes through the aquaporin, or it cannot let others pass through. Now there are also carrier proteins, basically the channel proteins that facilitate. These are channel proteins that facilitate the movement of specific molecules across the membrane. And in this case, the molecules diffuse down their gradient of concentration. So like an example of this is the GLUT1 glucose transporter. Basically, the outside of the cell has many amount of glucose if there's any unbound ones, but then if one goes in, it will be like, okay, this is happening. This is glucose, it binds to it. It's like, okay, let's change conformationally, that way the glucose can go into your cell. Now with active transport pumps, there's a different scenario where it uses ATP, which is basically an energy component. We'll talk about that in the next video when we talk about energy and metabolism. But primarily with ATP, what happens is that it allows the molecules to be transported against the electrochemical gradient. So with what we've talked so far, it goes down the gradient. Now we're going against it. So a clear example of this is the sodium potassium pump, or this sodium potassium ATPase pump. Basically uses ATP to transport sodium and potassium throughout. So for every 3 sodium ions pumped out, 2 potassium ions come in, and it ends up with the net decrease in the positive charge inside the cell, and the both ions are basically pumped against their gradient. So if there's like more, if there's more inside, no, if there's less sodium inside, it's pushing away from it. So it's going to where it's more sodium. Instead of going high to low, it's going low to high, basically. And this is kind of the mechanism itself, where we have the unbound protein here. Then it goes into, okay, the sodium, 3 sodiums go in, it binds, and then it's like, okay, we need ATP. ATP comes in and binds to it, and it's like, it changes the shape, the conformation itself of the potassium pump. And what happens is it releases the sodium, and then it's like, okay, now to get this up and running, we need potassium ions to come in, and 2 of them come in, they bind, then it releases the phosphate that was taken from the ATP, and it releases it, and then it releases the confirmation that it changed. And then it goes releasing the ions themselves, and that's how the sodium potassium pump works. And that is the end of the structure and function of plasma membranes.